Hi, this is Dr. Vojdani. I'm an immunologist, technical director of immunosciences laboratories. I'm going to talk about the importance of measuring IgG and IgM against three different viruses: Epstein-Barr virus, herpes type 6, and SARS-CoV-2, which are involved in more than 15 different autoimmune diseases. Our immune system has two different compartments: the innate and adaptive immunity. That collaboration between them results in protection of the body against foreign materials. This collaboration between innate and immune system is highly important in protection of the body against bacteria, viruses, parasites, and other environmental factors. Any malfunction in this collaboration between innate and immune system may result first in inflammation, autoimmunity, and other complex chronic disorders. Many environmental factors, such as food, toxic chemicals, and especially pathogens, particularly viruses, have or can have significant effect on this collaboration between immune system, collaboration between innate and adaptive immune system, causing malfunction, and this malfunction in the immune system results in autoimmunity. Viruses such as Epstein-Barr, herpes type 6, and SARS-CoV-2 are very small particles containing RNA and DNA with a protein coating. These small particles, the viruses, cannot survive on their own. In order to survive, meaning to infect and replicate and spread, they have to grow or infect human tissues or mammalian tissues, such as muscle, epithelial cells, heart, brain, and others. So these viruses, using the resources from the tissue in order to grow and spread. However, during this process of growth and spread inside the tissue, in order to survive the immune attack, meaning the innate and adaptive immune response against the viruses, they acquire new proteins. For example, when Epstein-Barr virus infect epithelial cells, they acquire proteins such as nucleoproteins, ROSM. And therefore, when the immune system will attack the virus, the body is going to make antibodies, not only against the viral particles, but the new proteins that acquired from human tissue, which can result in this situation in lupus. Herpes type 6 loves to grow on neurons, and during the process of their growth, they acquire a protein very similar to myelin basic protein, which is involved in multiple sclerosis. That's why herpes type 6 is involved in many autoimmune disorders. And finally, SARS-CoV-2 loves to grow on a variety of cells, and that's why SARS-CoV-2 is involved in more than 12 different autoimmune diseases. So this process of acquiring new antigens, the virus acquiring the new antigens, new proteins that are similar to human tissue, is called molecular mimicry or cross-reactivity. So when the body reacts to the viruses, making two kinds of antibodies. One, against many components of the virus, in order to bind to the virus and neutralize, meaning preventing the growth of the virus. But also due to acquiring new proteins that I explained before, the antibody produced against those proteins can cross-react with human tissue, resulting in autoimmunities. That's why it's crucial and important to measure antibodies against these three different viruses that are involved in autoimmune disease: Epstein-Barr virus, HHV6, and SARS-CoV-2. If IgG or IgM antibodies are elevated, then your practitioner may consider to order autoimmune panel, which includes anti-nuclear antibody, ENA or extractable nuclear antigen, rheumatoid factor, immune complexes, smooth muscle antibody, and mitochondrial antibodies. So it is very important to measure not only antibodies against these three viruses, but also to consider immunosciences lab autoimmune panel. For further information, please consult our website, and also two very recent articles, one of them which was published in Frontiers in Immunology, which I have it with me right here, about the title is Reaction of Human Monoclonal Antibodies to SARS-CoV-2 Proteins with Tissue Antigens, Implications for Autoimmune Disease, published on January 19, 2021. Highly recommend to read this article, and also another article which was published in Autoimmune Reviews under the title Serological Cross-Reactivity Between Viruses and Their Contribution to Autoimmunity. Thank you.